So the question one, supplementing one's diet with probiotics are common treatment for several diseases, such as gastrointestinal disorders or skin diseases. Could these off-the-shelf supplements have an effect on insulin resistance, or perhaps only some specific bacteria strains can be used for that purpose? Not all probiotics are the same, and respond to probiotics can vary in each individual. What works for one person may not work for another person. For example, specific strains such as lactobacillus and bifidobacterium species have a modest positive impact on insulin sensitivity. Probiotics may depend on the factors like the microbiota composition and health and diet of individual. So diet and other lifestyle factors play a role in the insulin sensitivity and insulin resistance. Can you hear me?
Yes, yes.
Okay. So question number 2. Is that visible?
Yeah.
Okay. Why do you think the proportion of insulin resistance was lower in individuals of cluster C? The cluster C basically belongs to insulin sensitivity-associated group, bacteria 2. The group 2 bacteria contains bacteroides, aristides, and flavoniferators that are negatively correlated with the majority of the carbohydrates. So cluster C taken here. This is yes, the proportion of insulin resistant bacteria in these clusters are more. So that's why they show the lower insulin resistance in individual of cluster C. The proportion of insulin resistance among individuals with abundant group 1 and 3 bacteria, which are in clusters A and cluster B and cluster D, was consistently higher than those group 2 bacteria which are present in cluster C. They identified on the basis of shotgun metagenomic data. So this is question 3. Why do you think interleukin 10 mediated only the effects of fructose, mannose, xylose, and rhamnose, but not other metabolites? So, the specific interaction between interleukin 10 and fructose, mannose, xylose, and rhamnose may be due to the unique structural properties or metabolic pathways that are associated with this specific metabolite. The response to metabolites can vary among each individual due to unique genetic architecture of the microbiome and other environmental differences. So these metabolites could potentially trigger the immune responses or modulate immune cell behavior through the specific receptors and pathways. Some cellular pathways are involved and are not activated by the other metabolites. Additionally, the composition of gut microbiota may play a role in the metabolite interleukin 10 interactions, and these metabolites may have a distinct impact on the microbiome compared to the other metabolites. This is our opinion, this is our opinion and our understanding. So ultimately, the specificity of interleukin 10 mediation for these metabolites like fructose, mannose, xylose, and rhamnose likely results from the combination of some molecular interaction, some cellular pathways, microbiome-related factors, and other metabolic pathways that distinguish them from the other metabolites.
So, I have a question about this, about the answer, number 3. So there is not such a thing as a direct connection already well known or well studied about the connection between interleukin 10 and these metabolites, right?
Yes. There is no direct connection.
Oh,
yeah.
Okay, thank you. Sorry, Dr. Owen, I hear you talking sometimes, but I don't hear your voice clearly. If you could, when you talk, if you could get closer to the microphone, please.
I'm basically commenting to Shazia how to respond. So when I will be talking to you, I will come in front of Mike. So don't worry.
Okay.
Okay, next move to the question 4. When comparing population with metabolic syndrome, pre-metabolic syndrome, and control, they found a big difference in xylose and feces. Xylose is a typical component of dietary sugar. The richer in sugar is your diet, the higher the amount of the xylose you might have in feces. How do they know this increase is caused by a different bacterial population in the digestive system rather than by diet high in sugar? So this, I think, to determine if the increase in xylose in feces is primarily caused by a different bacterial population in the digestive system rather than by a high sugar diet. So some researchers typically employ the several investigative strategies. They're working on it. In order to separate the effect of diet from the effect of gut microbiota, they conduct the controlled dietary intervention study. The analysis of gut microbiome using techniques like 16S ribosomal RNA sequencing or metabolism sequencing, and comparing the composition and diversity of gut bacteria from the different groups. It can also help the identification of potential bacterial candidates that are responsible for the xylose metabolism. And some other advanced statistical methods are also used to assess the relationship between xylose level and the diet as a specific bacterial population in the gut microbiota. So if an association is found between xylose level and specific bacteria, it may suggest a microbial influence. So some manipulation of the gut microbiota directly, for example, through the administration of probiotic or antibiotics to investigate the impact of specific microbial population on the xylose level. So in this study,
sorry, I have a question about the previous answer. But this is what researchers might do, but not what researchers have done in this paper, right?
Yeah, this is not in the paper, but they study, they work. In the study, Stipe Springoldi, when they administer this bacterial strain in the mice, they increase the xylose level in the mice.
Okay, but it's still...
This is mentioned here.
Mm-hmm. Okay.
Yeah, she increased the xylose level in mice.
But for example,
but the mechanism is not present in the paper.
Yeah.
What is the mechanism behind this? It is not present in the paper.
Yeah, but
just they administer specific bacterial strain to mice, specific quick feed diet to the mice, and then check the level of xylose and other metabolites. So towards.
Yeah, but still in this because I was surprised when I was reading the paper, because in no moment they consider or they mention that the diet of the people investigated, of the subjects, was controlled. So I was wondering, like, if this augment of xylose was really related to the microbiome, and if that's and it could be because of a different type of diet. But even if they found an increase in xylose in mice that have been administered with this strain of bacteria, it could be caused by many other things. It could be because many, for example, bacteria strains might not work individually, might work as a complex. Like bacteria A depends on bacteria B and depends on bacteria C, something like that. So I don't know how trustful or how relevant is that connection between those bacteria, those different bacteria on the gut, and the increasing of xylose in feces. I have doubts.
They have provided the quick fat diet to all of the mice, but they ingested a single strain to one by one in each mice.
Yes.
So based upon the composition of diet. So in all of the mice are using basically the quick fat diet, but there are only one Aristides fine Goldie mice. I mean mice that contains Aristides fine Goldie. The other one contains the Aristides indistinctus, and the same for the others. So using that data, they decided or they observed that the Aristides fine Goldie is basically responsible for increasing maximum xylose level, as far as I understand.
And did they do any experiment with mice where they supplied with the same combination of bacteria? For example, for the cluster C was characteristics for having like 3 different strands of bacteria. Did they try to reproduce that in mice? I think no, right?
No, we don't have any experience.
No, they don't.
Yeah. Okay.
And don't you think
They not.
and don't you think that perhaps that would have been more relevant than just like supplying one strand of bacteria?
I mean, there should be. I think there should be an experiment in which they should. You were talking about that there should be an experiment where they should provide all of these strains in one mice.
Yeah, for example, yeah.
Yeah, it should be there, but they did not perform such.
They did not perform this.
So
They perform individually.
Alejandra, somewhere in the paper they have generated data that different strains of bacteria, they are producing some enzymes. Those are extracellular enzyme of the microbes, which were basically catabolizing the carbohydrate present in the gut. So probably some genes are present in this particular strain of L-skips, which are breaking down the complex of the carbohydrate to generate xylose. So what that particular enzyme is, I think that needs to be looked at because glycosidase or fructokinase, or there are many carbohydrate degrading enzymes. Those were secreted by the microbes.
But
Those were influencing the monosaccharide levels in fecal samples. So this kind of experiment needs to be done.
these kind of enzymes were abundant in group 1 and 3 bacteria, but not in the mainly glucosidases and the fructokinase. I think these enzymes were abundant in the group 1 and 3 bacteria, but I think there was alpha amylase or amylase enzyme that were abundant in the Aristides fine Goldie that basically break the polysaccharides, but not further into the monosaccharide subunits. So that may be the reason that Aristides fine Goldie is responsible for the insulin sensitivity.
Okay. Okay.
Can I move towards next slide?
Yeah, you can continue. Yeah, thank you.
Okay. So the next question, fecal carbohydrates moderately explain the presence of interleukin 10. Is there any cellular pathway connecting them? So the connection between fecal carbohydrates and insulin and interleukin 10 involves some cellular pathways that are related to the gut microbiota, short-chain fatty acids, and other immune cells. So when gut microbiota ferment the carbohydrates, they produce the short-chain fatty acids, which can generate signal through the G protein coupled receptors that are present in our immune cells. This signaling pathway can promote the differentiation and activation of T regulatory cells and the production of interleukin 10. So additionally, the short-chain fatty acids can enhance the gut barrier function and reduce the inflammation and contributing to the interleukin 10 as an anti-inflammatory effect. So therefore, the cellular pathways connecting the fecal carbohydrates and interleukin 10 include microbial fermentation, short-chain fatty acids production, and G protein coupled receptors, signaling, and immune cell responses. So in this study, there is a cellular pathway that are connecting the interleukin 10 and so.
There is no mechanism to explain in the literature. Yeah.
So, move to the next question. In this section, insulin-sensitivity-associated bacteria in experimental models. They said that we detected 13 out of 15 carbohydrates associated with insulin resistance in the bacterial culture, the experiment. But how many carbohydrates were found in the total? Is 13 a relevant number in the comparison with the total number of carbohydrates found in the cell culture? So, here is the results. So they recognize metabolites in the bacterial culture, total 22 human fecal insulin resistance and insulin sensitivity associated bacteria. They culture 22 total. These bacteria were selected on the basis of findings from the genus level co-occurrence and the species level profile. So they identified 261 metabolites in which the principal component analysis plot of 198 metabolites indicate that the species Bacteroides insulin-associated bacteria order show a distinct metabolic profile along the PC1. So the top 10 metabolites contribute to the group's creation, including the several amino acids and fermentation products such as succinate and fumarate, and the majority of these metabolites were preferentially produced by this species Bacteroides. So they detected 50 and they detected 13 out of total 4 in their experiment. Yeah.
And those
Total they identified 50 carbohydrates that are associated insulin resistance.
And those 13, I understand they were just in this list of 261, or perhaps they were the ones that were more highly produced, or low, or was there any difference in terms of like quantity?
Well, Alejandra, in principal component analysis, there is separation on the basis of the variables or clusters which are separating different groups.
Yes.
So there were different, as far as I understand, different groups for amino acids or other metabolites. So they have selected the group of carbohydrates in which there were 15 carbohydrates. They were importing or contributing for this separation in those bacteria. So based on those 15 carbohydrates, once they tested in the culture medium, they have found 13 of those were then strongly associated in the later experiments.
Actually, and they identified insulin resistance and insulin sensitivity too, both detected in the overall paper. Then they shortlist some 15 carbohydrates that are associated with insulin resistance, and they use for further analysis.
And yeah, okay. So that's for the 15 carbohydrates. But what about the 13 carbohydrates? The 13 comes from the list of 261, right? So those 13 were, for example, highly expressed, or they were less expressed? Because you know, sometimes when you have a list of genes or a list of like, in this case, metabolites, some of them are expressed just like symbolic, almost like very low expression, and you don't know if that has really an effect and a phenotype, and others can be very highly expressed. So I was wondering if perhaps it was written that those 13 was highly expressed, or they were relevantly expressed, or if you knew.
Actually,
They were highly expressed, that's why they are responsible for the
insulin resistance.
Okay. Thank you.
And they are differentially expressed by different bacterial strains.
Okay.
So next question, I need explanation about this. Then discuss in the paper the analysis and the interpretation. So Fazan explain it clearly, I think.
Hi everyone, can you listen me?
Yeah.
So actually in this graph they have plotted when they compare the two variables. So actually the area under the curve will predict the difference, or you can say determine the overall dependence of the two variables on each other. So area under the curve, for example, if we see here on the y-axis there is an area under the curve. So in area under the curve, if you have a maximum threshold value, then you can separate your value as possible as, because if you look, for example, if we have a false positive result and some false negative result, so you have to differentiate in a cluster or in a group. So on that basis, they have designed a area under the curve graph. So they have selected over different number of selected markers on the y-axis, x-axis, and then plot their graph on the area under the curve. So for example, if we see over here, we have 4 different type of variable that include 16S, orthodox, metabolome plus orthodox. So they have compared all these. So the major threshold value for that one is more than 0.8. That means there is a possible that we can differentiate the overall selected markers that are differentiated from each other. For example, you see on the metabolomes, so there is a maximum value or maximum peak on the 10 variables. So there are 10 variables that are maximally differentiated from each other. That there is a minimum chance of the clustering at between each and other. There means there is no false positive result in that. Followed by that, if you look on the metabolome and the carbohydrate, then there is a same overlap graph on the metabolomes. So that is showing that there is a same threshold over that value. Only that 10 selected marker or more than 10 selected markers are considered as the differentiated, or there will be some sort of a good indicator, or you can also say that has a good statistical value or a good statistical analysis for the further analysis. So if you look in that graph, the various scattering data is showing. So for example, if you will...
Hello, excuse me. Can we get back there?
Yes.
Just the previous slide. It was actually my question. So can I'm now trying to share my screen. Do I share it or not yet? Not yet. Okay. Can you stop sharing, please?
Yeah.
Okay, let me share my screen. Is it sharing?
Maybe you're sharing there?
Yeah, it is. Okay, that's great. So then let me get back to here. Okay. So what can I just stop here for a moment, please? Nastya, Donfa, Alex, do you have any questions? Did you understand like the explanation about what is the method, what is the analysis type, and what is the kind of what is it showing? Like, do you understand this?
I didn't stop so much to analyze the picture. Like,
you didn't
so deeply, yeah, no.
But not do it. Like, what does it mean? Do you understand the whole?
I didn't understand.
Yeah. Okay. So, Donfa, what about you? Okay. Sorry, we still don't understand. Like, what is going on here? So, excuse our ignorance. We are not really familiar with this type of analysis. So we need more explanation there. So, so what is, first of all, what is 16S means that they make the analysis of different genomes of different bacteria based on the different sequence on the 16S. Is it correct?
Yes.
Yes.
Right. KO means that they analyze the orthodox based on the KAGG, right? KAG database.
Yes.
Basically, they are trying to find the similarities based on those orthodox between different bacteria, right?
Genes.
The genes, of
They
course.
form
Orthologs of the
yeah,
genes.
gene.
Orthologs of the
Yeah.
same genes in different bacteria. Is it correct?
Yes.
Right. Okay. And then like the metabolome and metabolome plus KO, like what does that mean? Like what are these two additional groups? I don't quite understand this grouping. Like they are trying, what is the take-home message here? Like what they're trying to compare here? I don't quite understand that.
Actually, they are comparing a predicted gene and metabolome relationship between them. So initially, they predicted the 16S gene sequences, then compared with the cat ortholog between the different various other bacterial species. Then they analyze the metabolome data from the GCMS. So they own that the metabolome data. Then they correlate with the cat orthologs. That how they can correlate the other metabolites that they have predicted from their own data that from the GCMS.
Okay, I still don't understand that. I don't understand like what is on the left we have the area under the curve of what exactly? Of area under the curve of what? Firstly, secondly, on the X there is number of selected markers. I don't understand that axis either. So as I said, so excuse my ignorance, but I think my lab mates also don't understand this fully. Can you please explain, like, if I would be a 10-year-old child? Okay, so like, what is it about? What is this? What are they trying to show here? What do they compare?
Actually, they are comparing the metabolomes and the associated genes. So they selected the different metabolites, or you can also say as a marker over here.
Okay. So the curve metabolome and metabolome plus KO relates to the metabolome, right? So they make metabolic profiling based on probably mass spec, right? Of what? Of feces, of those people, right? They took all these people with and without insulin resistance, with the metabolic syndrome and so on, and they made they profiled their metabolome, right? So then, like, what do we see on this figure A exactly? So the curve, this metabolome curve, what does it represent?
I think I understand.
Okay.
I think I understand. So they are like comparing the metabolome and all the different genes expressed, and they are comparing like many of them. For example, they compare 50 of them. So if there will be like 30 of them will be different, they will be from 10 or whatever value from here, but it will be from the value 10 to the value 30, it will be a difference. But actually all of these are the same. So there are only 15 of them are different, because there is only like a difference of a slope in the first 15.
Oh, I see.
And then this value, I don't know exactly what it means.
Yes,
but
let's ask, because like we have professionals here, so who can explain what is the area under the curve? What does it represent? And
So the area under the curve actually define the threshold on which you can say that how your value is properly sorted. For example, if you have one obese person and you can sort in that this person is not obese, so there will be some threshold value that the area under curve is showing the threshold value, that is a point A.
Wait a minute. You remember, you're trying to explain to a 10-year-old child. I think the 10-year-old child would have problems to understand the threshold values and so on. Like, what is exactly this area under the curve is under which curve? Like, which curve are we talking about? Because we don't see the curve here. And if a child is being told that he has an area under the curve, he would like to see a curve. Where is the curve? They don't show the curve because it is probably too obvious for them, but it is not obvious to us, 10-year-old children.
Actually, if you see in a simple example, I will say that there is the shelter beside where we are living. So this all shelter define the overall the value that how you can live in certain area. So if you consider in such a way, you will easily understand. For example, if you would say that 0.8 is a threshold. Threshold is a value that is defining that from above this point, your data is reliable, or you have sorted the data in a very well positioned or in a very well mannered.
Okay,
And what is the value? What is that value?
What is this value?
04
exactly?
or 6
Is it,
or 8? Which
That is for
are those the peaks
that is for that
on the mass spec?
that is
or-
for metabolism. That is for metabolism is about more than 0.8 for metabolism. And 6 for the 16S is about 0.6 around on average.
Sorry, I still don't understand. Is the area under the curve is it area below the peak on the mass spec, or it is something else? I still don't quite understand this.
It is the area under the curve. Actually, area under the curve is representing the whole area that is behind that. So
There is not a specific curve, there is an irregularity. if we see. So they will mean calculated, I think, all the area that are under the different curves. Like if we see the last one, the lower one, line is wrong, okay. The lower one drops. I mean, there is an irregular. I mean, first there is an increase in the value, then decrease, then increase, and so you can see that there is an irregularity. So they calculated the overall area under all of these curves, and then they decided that which one is better.
I still have a problem to understand what does the curve mean. You say there is no specific curve, but there is some area. Area means there should be some kind of area which we can see, which we can visualize. And now I don't really. So I don't fully understand what is this area under the curve, as I said. So excuse my ignorance, but I don't. So what I understand from what Alex told me is that we are looking at this particular part, these 15 metabolites, which are different from the metabolome and the metabolome plus KO. That is, and we find that it is interesting because that is the only difference where we see that there is some difference between which is an added difference based on the orthologs from the CAC, right? And this is interesting, right? So the take-home message from the A is this part, this initial part between the metabolome and the metabolome plus KO. Is it correct?
Yes.
Yes. This is basically statistical explanation of the data spread and how much, as far as I understand, we are not expert in this data analysis, but as far as we understand that this is multifactorial data from different experiments like 16S data and then the cake ortholog data and then the metabolome that you are referring that under the peak of MS. No, that is not under the peak of MS. Basically, that is the metabolites, whether those variables were 10 variables, 20, and if you have 20 variables and then you have 30 and so on variables. So, which number of variables will you rely most on which of the data set? So they have plotted this area under curve, and there is a related technique called receiver operating curve, and then that is statistical representation of the data, that how you can rely whether the 16 sRNA data was good and whether the metabolomic data was good. So if you have 10 data points or 10 markers in the 16 sRNA or the metabolome, then you can see the difference in the first place over here. And then if you have markers, so if you have more markers, 40, then these 3 data sets, they merge together. But this one is still coming with less priority. This is not that much reliable as much as these data set would be reliable as far as we understand. So this is, I understand, difficult to interpret and difficult to explain graph, but that is basically it.
Okay. I should admit I still don't understand. I don't fully understand like what is the purpose and what is the method behind this analysis. So maybe we should leave it by that. But I mean, I understand that this figure on the right side, it is.
You're not sharing the screen now.
Oh, okay. Why is it not? Okay, wait a minute. I'll try to share it. So I understand that the figure on the right side is representing the quantification of the data on the left side. Okay, this I understand. But my point is that I don't fully understand how exactly and why they did this first figure, but it seems like a key figure to move forward. So I think I have a problem with Jitsi. I cannot get into the Jitsi.
Again, I would say again that is indeed difficult to understand and explain. But as in normal case, whenever we sample different, say for example, 100 humans from different locations, then we generate a curve that is whether that is normally distributed data, bell-shaped or skewed. So probably they are representing their data, that how much you can rely on which data set and with which confidence.
Okay. So probably we can leave it, but
someone with statistical background, he or she can explain it very well.
Okay. So so as I said, I still have my questions there. I simply don't understand how did they come with this data, and as soon as I don't understand how they come with this data and like what they really want to tell me by that, yeah. So I have problems to understand the whole concept. I mean that's my problem, but that's just my problem because I'm not an expert here. So I'm trying to join using a different browser. I don't know why it's not working on the. Okay, let's see. Join a browser. Okay.
So. Can I go to the last point?
Yeah, sure. We can maybe later join again to discuss, because I have many questions like that, so where I don't fully understand the design, and this per se kind of raises questions about the interpretation. But okay, sure. Go ahead.
Okay, so using this technology. Using technology like omics technology, it explored the involvement of microbial metabolism and their products in the insulin resistance and insulin sensitivity. So with the help of this study, we can also identify the gut microorganisms that are involved in the carbohydrate metabolism and that influence the obesity and pre-diabetes.
Sorry, it was me.
So in the study, with the help of this study, you can also identify some species, bacterial species or strains like Ascarius indistinctus, that is basically gut microbiota that improve the whole body insulin sensitivity and its administration metabolism and thereby insulin resistance. So while simultaneously reducing the intestinal monosaccharide level. So fructose is a widely recognized risk factor for inflammation, insulin resistance due to its population. It also highlights in this study excessive monosaccharide have the potential to promote the ectopic lipid accumulation while also activating immune cells and leading to the low-grade inflammation and insulin resistance. So this is the overall conclusion of this study, and do we understand? Okay. So we can continue for further discussion. Thank you so much for your attention. That's all about your questions and comments.
Okay, would you like to make some kind of remarks overall about this study, your own opinion, or some critical points, or anyone, before we get back to the more detailed overview?
Yeah, it was interesting and very challenging paper. So it's very difficult to understand, but I try for my best and I try to understand, which I understand. So this is the overall overall take-home message, and this is the explanation of this paper that I explained in my question answer comments and in my previous PPT. Yeah, it could be different and interesting treatment for obesity and several non-communicable chronic diseases.
Okay, anybody else would like to comment anything?
I mean, overall, to me, the paper was not very convincing. I think the connections that they draw, like you have this type of bacteria, and then you have this feces, and that is related. I didn't see a clear connection on that. I mean, it could be, but it could also not be. It could be that you need several strains of bacteria. It could be that the patients were having different diets because it was never assessed and it was never controlled. It could be, I don't know, it could be so many things. I think the only values they evaluate to sort the groups was like if they were pre-metabolic syndrome or they have metabolic syndrome, or if they didn't. And also the body mass index, I also saw they measure that, but still without a diet control, I think I have many concerns. And then of course, because they didn't prove any type of mechanism, I'm also not convinced. Yeah, and I don't know, like bacteria in general, they can produce many things. Monosaccharides is a thing they produce quite a lot. Bacteria as a disposable matter. So I didn't see the connection clearly. I understood the paper quite well, but I didn't see a solid connection. Those are my thoughts on this paper.
Yes, these are quite valid comments, Alejandra. But since these complex diseases are always complex and multifactorial, so these are difficult to have controlled experiment in real life situation. So whatever in everyday people they are eating or they are doing that, they are assessed based on that. So what the researchers have done, they have included the participants who came for the regular medical checkup, just to avoid the confounding factors of the chronic disease, whether they were diabetic or with other chronic disease. They excluded that, and they only include in the pre-diabetic condition, they were like pre-metabolic syndrome, and even their BMI levels were quite stringent for Japanese population. Those were not applicable to European or American population. So, keeping in view your comments, those are valid, but what could be done in such situation? They have done in a reasonable way, and they have included different data sets from the host metabolome, then the fecal metabolome, serum metabolome, and then the fecal metabolome, and then the microbial identification, and then they made a transition based on strong statistical evidence, whatever they have got. And obviously, such kind of studies need to be replicated in large number of population in the same population, and these results need to be validated in population as well. That sounded an interesting study to us. Thank you.
So,
Okay, wait a minute. I will try to join now with
So let us do this.
yeah, I will do it. Okay, so I'm yeah. Can you hear us?
okay, please continue. Please continue.
Yeah. Okay. Okay. I'm trying to find the paper, which is from. What is this? Oh yeah. This. What was the name of the paper?
It has a title.
Here. Okay, maybe we can open it on a different on that device,
On my computer?
or they can open I'll try to open it. Okay, Nastya, Donfa, do you have any other questions or your maybe problems or anything like which you would like to address here to in regard to this paper?
I didn't fully understand the methods that they used and these some diagrams, so I don't really grasp the whole idea of the study, and the results were very not very convincing for me because I don't understand how these connections were made.
Okay, let's try to get into this, and let's try to find out, and let's try to address those concerns, because I also have some concerns as you see. So let me share my screen. Okay, I hope this will work now. Okay, can you see the screen? Okay.
Yes.
Okay. So let's just do this. Like that. Okay. Okay. So. Okay. Like the figure A, the panel A, I still don't understand this. How, like what is the take-home message here? But usually they write it in the results or in the discussion. Like what is their interpretation? If Shazia can tell me like what is in just one sentence, like what do they show by figure A? That would be great. Then we don't need to maybe go to the methodology. Maybe we need to study it separately because it would take too long time. Because we already tried to understand, and I still don't understand how exactly, what do they plot on the x, what do they plot on the y, what is exactly this area under the curve? All these questions, they are still unresolved for me. So that is the first thing. Okay, anyone wants to make a summary for me for this panel A, please? Because as I said, so I had some problem with that.
And I will try if I could make it clear. If you take the example, if you take meat to food, and if you want to categorize those food that either it is good for the diabetic or not good. For example, if I'm talking about rice and wheat, so which one is much better for the diabetic? So in that case, we usually categorize these food on the basis of their glycemic index. And for that purpose, or in order to find out the glycemic index, we basically measure area under the curve at different time points for that specific food.
Area,
and
sorry,
we compare.
sorry, area under the curve of what? Curve of what? Curve means like there is some kind of curve of abundance or what? Abundance of different metabolites? Or?
Yes. Yes. At different metabolites or at different time points. Whatever the study design is. So here it is about, I think, about the metabolite that we have 10 different metabolites, and they are basically trying to find out area under the curve for that metabolites. That what is the total area under that curve? That could be possibly a single curve. That they increase and then decrease. So there is an irregularity.
Sorry, Shazia.
Yeah.
I don't understand what is the curve here. Like that is the problem. You say you're talking about the area under the curve for a specific metabolite. But in order to have the curve, you need to have like what is on the x if you have a curve? It is time or what exactly? And what is on the y of this curve where we measure the area under the curve? Is it the abundance of this metabolite?
It's different.
Difference of what?
I think it's difference. You have like for example 50 metabolites, and here you measure how different they are. I think, and then because there is like a slope, it means there is a difference. If there is no slope, there is no difference. But I don't know what the values are. I think that's the main.
There is an abundance of these marker along x-axis, and along y-axis there is an area under the curve. From starting up to the last point, how much area is basically taken up by these points.
Okay. I still. No, I don't understand. As I said, so I asked like, okay, if there is anyone who can explain it to the 10-year-old child, then in simple terms. Like here, I don't understand what the area under the curve means. Still, usually all those kind of mathematical concepts, they can be still explained in more simple terms. Like here, I don't understand the simple idea of that. But okay, maybe we should move on with this. Okay, so for the B, as I understand.
I have a very simple example, but that is somewhere in my computer, so I could not retrieve it right now from my laptop. So but I could explain that. If you could have time, we can discuss it later as well. By taking some data that I have in a very simple way, or in the form of the single graph, so I could explain that to you. Will you appreciate it?
Okay, maybe we can come back to that later. I don't fully understand the method behind and like the analysis, but what is the take-home message, at least from the A? In one sentence. In one sentence, what is the take-home message?
What we could do that we could make a simple video for almost 5 minutes that we could share with you later. So from here it is I think difficult for me as well to make you understand.
Oh wait. I'm asking what is the take-home message? Like one sentence. Take-home message from the panel A. How you understand this?
One sentence to take home message. Thank you, Nakanishi. Again we got.
इससे क्या ये तो मतलब वैसे स्टार्ट के वीडियो मेटाबॉलाइट्स को दे रहे हैं स्टार्ट वो ये ना कि मतलब क्या क्या करना है मतलब इसमें से मेटाबॉलाइट्स में है कि मतलब इन सब को आइडेंटिफाई
I think they try to make us understand that which data is better to proceed and
this is a selective data that is used for analysis के लिए I think this is the selective data that are used in this oral paper.
Huma or Shazia, can you please make just one sentence take-home message from the panel A that we can just take as a like like the cornerstone to move further.
They've just filtered the large data and used some of the parameters like seen as some metabolites and that they used for further studies. So they just tried to show that data in these two graphs for further analysis that they used.
Do you understand this? I don't understand this. I don't understand this take-home message. You say they use some kind of they analyze different data and they try to find the way like which data are different. I mean this is kind of I don't quite follow.
Hello?
Yes.
You are asking about what is the area under the curve actually?
No, I'm asking about the take-home message for the panel A. One sentence take-home message. What does it show? What does it tell us? Each item, each figure, each table has
defining the sensitivity and the specificity. If we simply talk, it is defining the sensitivity and the specificity of the specific selective marker. If we will specifically talk about this.
Okay.
Because the area under the curve is especially designed to define the selective specificity and sensitivity of the data. So that is the area that we are defining. So the threshold value that I am talking earlier about, that was actually the threshold to defining the specificity and the sensitivity. Over that point,
sorry. Excuse me. Yeah, you already tried to explain that. But for me, in order to really see like what is it going on, I need to see like how is it done? How is it this area under the curve? How do they get this? And like I know they can write about specificity and so on, but without I cannot believe them. I need to see like how exactly do they do this. But my question is now not about the method and about analysis. My question is what they want to tell us, because each figure, each table in the paper is telling us something. So what is their message? Can you tell me in one sentence what is this panel A telling us?
By using this data, they can predict the insulin resistance basically. By using this data.
Okay, how?
They use different methodology, like you can see 16 ribosomal RNA can go through the metabolomics data of fecal samples and and they compare the metalomic and cathode log together. So
initially in this data, they simply compare the metabolomic syndrome and the insulin resistance person. For example, if you see the graph A and the B, there is a participant that are over by the fecal concentration.
Yeah.
For example, the fructose, and then they analyze the different parameters that are associated with both metabolic syndrome and insulin resistance.
No, that is the initial goal of the paper. I understand that, but I don't understand the specifics because I don't understand how exactly they come to this. But okay, maybe we should not stay here on this panel A for too long. By the way, how do they define metabolic syndrome? Because they, like some different clinicists and different labs, they define it differently. How do they define metabolic syndrome in this paper?
So in metabolic syndrome, they define that dyslipidemia.
Only dyslipidemia?
No, not lipidemia, the lipidemia, elevated blood pressure level, and the impaired fasting glucose.
Okay. Okay.
With the BM, with specific circumference of male and female.
Okay. Okay. So then in this figure B, they clearly show that there are some associations with these different metabolites, with this either metabolic syndrome and insulin resistance. This is quite clear, right? And then in the figure C and D, they are looking for, in the C, I guess they look for some correlations, right? Or
yeah. They find out the concentration of
the concentration of carbohydrate metabolites.
Okay. Okay, and this curve, this blue curve, looks like the most correlated, right? So this one, which is what? Which is fructose, right? Because I mean this way of showing the data where you don't quite nicely can distinguish the. Can you distinguish the colors of fructose and xylose? I cannot do. Okay, it's not so easy. But okay, it's xylose. Okay, they say the xylose, or it's called xylose, right? In English, I think that's the right word. Xylose is having the most predictive value. Is it correct? So that's what they say.
Yes.
Right? Okay, the take-home message from the C
right.
is a xylose, correct? Which is like most profound difference. And then in the D, they show, this is very strange way to show the significance values. It's not, I mean, what do they compare with what? No met with met and pre-met. You see those kind of, I think they always compare no metabolic syndrome with pre-metabolic syndrome and metabolic syndrome with only this control one. And then they say that there is some difference. So they can predict whether those patients have this kind of condition based just on the concentration of those different metabolites in their feces. Is it correct?
Yes.
Yeah.
Yes.
Okay. Now more or less it's clear. Then they find this. They made this clustering of different bacteria, right? Based on different stages, and they find the cluster C is kind of the one which is like having less incidence of this metabolic insulin resistance, and the other clusters they have more. And by that they found those bacteria which are probably, as they think, they are predisposing to having less incidence of this insulin resistance. Is it correct? Is it right how I interpret the panel A? Yes?
Yes.
Okay, and then they move to B, where they look for specific parameters, and then they always compare it with this cluster C. So they consider that cluster C is the one which is of non-metabolic syndrome, kind of a control, and then they always compare with this C cluster. So all the significances are compared to this third group, to the cluster C. Okay, so that's important. Okay, and we can see that indeed we can see the differences, and then the figure. Then they go to figure C, which you don't discuss at all, which is kind of again some kind of clusters, group 1, group 2, group 1, group 2. These are probably these groups of bacteria, I guess. So group 2 is probably this bacteroids and fecalibacterium, I guess. This group 2, right? Which seems to be protective, right? Because it looks like it is mostly elevated in this cluster C. And we see that it is really separated from other groups.
Yes.
But whatever.
Because this group is basically.
Yeah.
Yes.
Mm-hmm.
I'm saying that this group is basically consists of cluster C bacteria that are beneficial kind of, because these are involved in insulin resistance, and we can see that in the group 2, these bacterial group is basically linked with those metabolites that are lipophilic in nature, that are not linked with the carbohydrate based or other hydrophilic metabolites.
Wait. But how do you see that? I see.
While the rest of others, group 1, group 4, and group 3.
Yeah, I see. You mean that group 2 is most interesting because it is protective it is against this kind of insulin resistance, right? And it is separated from other groups, we can clearly see, and we see those blue triangles that they encircle. And those sky blue triangles seem to be the most interesting ones, but what are those? I don't see the explanation. Like, what are those blue triangles?
If you just zoom out this graph, you can see that these blue triangles, these are basically the bacteria that are present in the cluster C, and the rest of like squares, they are differentiated in different colors, and it is written here that the red one is basically responsible or linked with the carbohydrate based metabolites. Then short chain fatty acid, the yellow one is also lipophilic or lipid metabolite. And then the green one, these are the other hydrophilic metabolites, and the purple, the last one.
Yes,
these are also the lipid metabolites.
Yes, but those are sky blue, and I don't see the sky blue here. Like that is my problem. They have their legend where they have this red, some kind of beige, kind of yellow, kind of greenish, but they don't have this kind of sky blue. So that, like, what is this sky blue here? I don't understand. These triangles in group 2. What is this?
Yes, I'm just trying to explain that inside the circle, group 2, group 1, there are different, these are the triangular, but these are different in colors, and they are basically showing the bacteria.
Okay.
And the spheres are already given, yeah. That is written here.
Okay. Okay, so what I want to say, I don't understand what do they want to say with this panel C. I see that this group 2 is very separate, but I don't understand their take-home message here.
If you just look at the graph C and D, you can find that here the sky blue, and in the graph D there is the long bars that are given in the blue color as well. So those are also basically the group C cluster bacteria. So this color is basically differentiating the bacteria into different clusters, that from which cluster they are belonging.
Okay, and now if we go to the figure E, which I also don't understand, so this is basically they're trying to correlate those bacteria and those different metabolites based on the abundance in the feces. Is it correct? Is it the right way how I understand? So the bigger the circle, the more the correlation, and the redder the circle, there is a positive. The bluer, like the blue one, is a negative correlation, right? So, and from here,
Yes, right.
and from here we see that like whatever is like what is on the very leftmost column, these fecal carbohydrates, where you have like from the dark blue towards the very red, right? So this is the correlation with what? With insulin resistance, or what is the criterion here? So what is the most interesting corner here to look at in the panel E? So I understand that it probably is either the very top row or the very bottom row, right? I guess, because those are like mostly correlating based on these fecal carbohydrates. Because as you can see, this whole table is sorted based on these fecal carbohydrates, from the most red one towards the most blue one on the bottom. You see that, right?
Yes.
And I think that is they sorted it that way because it made sense for them that the most interesting are either in the top row or on the bottom row. So what is the take-home message here? I also don't quite understand. So apart from that, for the top row, we say that phosphotransferase system is the highest correlated one with those with all other clusters, but not the cluster C, right? And actually cluster A. So this means that it is totally absent in cluster C, which means that kind of this phosphotransferase system seems to be detrimental, right? So that's what we can probably say, right? Is it correct?
Yes.
My interpretation is correct?
Yes. Yes, it is that.
Okay. I should say, like the way they structure this paper and also like how they illustrate this really it's great for the specialists in this area, but for non-specialists, I mean, it is very difficult to understand. Okay, and then in the end they go for this figure F, which I also don't understand. Figure F. What is this K01193? And so those are probably specific enzymes, right? Those are specific enzymes.
Yes, it is specific for different enzymes, and it is given here.
This I understand.
Expression number.
But this should have a specific a connection with what they showed before. So logically, I could understand that probably they use this phosphotransferase system, and then they look for specific enzymes which are involved in this phosphotransferase system to show that indeed, even when they go for the specific enzymes, they see upregulation in all other clusters compared to the cluster C. Is my interpretation correct?
Yes.
Okay, that's great. Okay, then let's move further. In the figure 3, they show this kind of hairy map of interactions of everything with everything. And what do they want to tell us? They want to tell us that the more silos we have in the feces, the more insulin resistance it can predict, and this is what we already saw that in the figure 1, right? So it is not very, it's not novel. What is novel is that they show some kind of links to different cytokines, because they have also done multiple different omics analysis. So the value of this panel is to show that there are different omics data and they are trying to correlate. And what is interesting is that this
Yes.
interleukin 10, right? It has multiple connections with
Yes.
those metabolites which are predictive for this insulin resistant state, correct? Okay.
Yes.
And what else? What else is interesting? Like we need to look for many connections, right? And since it is also about the microbiome, there should be also some connections to this left corner on the bottom, as well as in the right corner on the bottom, the associations with different bacteria. And from here, do we see anything interesting except for these bacteriolites, which are already shown to be implicated? And maybe this Blautia, Doria, right? So, and by the way, these connections, does it matter like what is the color? Because some are gray, some are red. Red means they are positively correlated, right? And the gray, they are negative. Gray are negative correlated, right? Okay,
Yes.
that makes sense, right? Because we see clearly that those metabolites which are highly correlated with the insulin resistance, they are negatively correlated with those bacteria which are on the bottom left. Okay, so that is making sense. Okay, and then they go for this B and C here, and they show that there is a very high correlation with the plasma metabolites and interleukin 10 and the fecal carbohydrates, right?
Yes.
And then they go for the C, and then they are trying to, like, this is really difficult to interpret, like how they make this kind of graph. So what they want to say is that xylose is highly correlated with interleukin 10, and interleukin 10 is highly correlated with this insulin resistance, BMI. So what they want to stress is xylose, interleukin 10.
If you just focus on the cytokine column, just look at the cytokine column, and then on the left and right side. So the cytokine connections towards the clinical features are towards the metabolites. They are showing with the which metabolites they are correlating with, or which clinical features they are correlating with. So if you focus on that.
This I understand. I mean, for the A, B, C, we see like the multiple connections, correlations, and so on. But you know, for me, the kind of take-home message where multiple metabolites they correlate with multiple cytokines, and which in turn correlate with multiple states. I think this is kind of like stating that there is interaction of multiple things with multiple things, which is kind of, for me, doesn't sound like anything, you know. So I like, there are many papers like that where like they have show these hairy interacting schemes where like there are multiple interactions. So for me, it would be interesting if they go for like specific ones later, and they can prove that indeed, so like what those correlations are indeed, they can be proven. So what they want to tell us, I guess, from the by the figure 3 is the importance of interleukin 10, the importance of xylose, I guess, and also that they are kind of like really correlated with those insulin resistant states. I guess that is what they want to say, because otherwise, if they tell that many cytokines and many bacteria and many metabolites they are interacting with many of the others, then it is like saying nothing, you know. So it is too broad.
You're right.
Yeah. Okay. So then, if we move to figure 4. Then what they in the A to say that.
In this cecal metabolites, cecal concept, in the line of the cecal part.
What is this? Okay, they look for the blood glucose in what is AI, AF, and BT? What are these ones?
These are the applications of different bacteria.
Styles indistinctus and styles Pingoldi.
Oh, those are different bacteria, right? So here.
Bacteria. This is the control. Control group, and this is the.
Oh, so here.
The blood glucose level.
Yeah, and they check the blood glucose level when they administered in the mice.
I see. So
with a quick fed diet.
Okay, so is it correct that they first? Is it correct that they withdraw the mice from all of the bacteria by treating them either with multiple antibiotics or using like totally sterile conditions, and then they inoculate with a specific strain of bacteria, right? So they make the controlled experiment where they have only one species of bacteria which they use based on those
bacteria they remove from the human fecal sample, then culture in the lab. Then after this they administered in the mice.
Wait. But before administering in mice, they should make those mice sterile from all other bacteria, right? Did they do this?
Yes. Yeah.
Okay. So this for me, like it's a clean experiment, right? So where they look for the specific interaction of specific bacteria.
It is not mentioned in the paper. They just take the 6 week old mice in the experiment, and they give the high fat diet before 3 weeks of the experiment, and then they administer the bacterial species with high fat.
Okay, interesting. So they don't need to withdraw any other bacteria. They just use those bacteria as the way to treat those metabolic syndromes as a kind of treatment on the background of all other bacteria which are there, which is making it even stronger. It makes this point even stronger. That is interesting. So and.
That why this is very interesting strain like L-types Fingoli.
Okay. My question: those AI, AF, and BT, can we point them in this figure 3A? Is it this all types, bacteroids and flavonifractor bacteria? Which are those? Can we find them in this scheme?
Actually, these are species. So actually, genus bacteroids.
Insulin-sensitivity-associated bacteria. Basically, these are these species.
Okay. So all of them, all 7 of these bacteria were from this left bottom corner of the figure 3A, correct?
Yeah.
Okay.
We are not sure about the
but it would make sense, right? It would make sense. Okay, and then they find that indeed 3 of them they can downregulate the glucose levels in these high-fat diet treated mice. That's very interesting.
Yes.
Okay, and
that's what they are not educating that there could be new treatment options for insulin resistance or diabetes using such kind of microbes.
Yeah.
Okay, and then they use the same 3 species, and they are trying to look at this insulin tolerance test. And here again, they use like very, very bad color scheme. It is difficult to distinguish this color scheme. And then they say, and then they say talk about group p-value, which is, for me, it doesn't make sense. Why are they talking about group P? Means like there is some significance somewhere. So you guys do go and just think whatever you want, like which exactly group is significant against which. That's what they tell us. They don't tell us specifically. Dear authors of this paper, I maybe you will be listening to this, but I really don't understand what do you mean by group P. Like, we are interested to see what is the difference between AI and the vehicle. Do we see really some differences between this blue group, the AI, and the vehicle? Is it significant or not? Can you tell us specifically and directly? They don't tell us that. They just say, okay, you just do it by yourself, probably somehow. Like, I mean, this is not the standard which I would expect from Nature. I'm also really surprised about these Nature reviewers that they let it through. I mean, this is not acceptable. Okay, but then we go to the C, where they look for the area under the curve. Okay, so that's where they look for the differences, and in D they find that the blue is of course significant, and even green is significant. Okay, then maybe I can take it back. That's okay for me if they show it in C. Then we go for the figure D, and there they look for the correlations of what exactly?
Yeah.
This is the same.
Yeah. Cecal metabolite, basically.
Cecal concentration of what?
Correlation.
Cecal concentration of fructose. Okay. So they are looking for the predictive value of fructose, glucose, and mannose in order to show the how does it predict the insulin tolerance test, and I guess this would be already in where, in the patients, right? Or where? No, the correlations between the AUC and insulin test. They don't say where. Is it also in mice? Is it also in these mice? And if
it does.
And that's the last figure of the whole paper. Why do they finish with this one? I don't understand. What does it have to do with everything which we saw before? Why fructose, glucose, and mannose suddenly, and not xylose? I don't understand that. Do you understand that? Why didn't they use xylose? They were talking the whole paper about xylose, but they don't talk about that here. I mean, we can clearly see that there is a correlation of those carbohydrates.
These monosaccharides are, I think, responsible for the insulin sensitivity or insulin resistance. Xylose is pentose sugar, and that is, I think, not directly involved in enhancing the blood glucose level, or that is directly linked to the insulin resistance. As far as
well, okay, so all in all, if you look at all of the data which they presented, this looks to me like a descriptive data paper. They show some correlations. They look for different metabolites. They make different omics. It's like then where you don't have any hypothesis, and then you just use this hypothesis less kind of paradigm where I can do omics. I can do omics here, omics there. I can do like this multiple omics. I can do these correlations, and I can see what is going to happen. Then they come up with this kind of different. They didn't go much into these enzymes. They didn't look into this bacteria. They didn't look into why those bacteria are having this different effect. Is there some kind of enzyme? Is there some cluster or operon? Is there some operon in this bacteria which is absent in different bacteria, which is helping to metabolize somehow? They don't go for any mechanistic studies here. like the only thing
Yeah.
interesting which they find is that okay, they can rescue this by these bacteria, but they don't even show how exactly does it happen. Totally descriptive paper published in Nature. I'm surprised, really. I'm really also, and
Because these papers nowadays get published very high.
yes,
Because
that's a
tons-
mainstream.
tons of million of
That's a mainstream.
For
That's the
the-
mainstream of research. They do okay. I do this because I can do this,
Yeah.
not because I have some hypothesis about like what could be there behind, but they just because I can do this because
Yeah.
the other guys did that exactly like that. They look at yet another paper in Nature or in Cell, and they just do exactly like that, but with a different setting. This is my interpretation. And they don't even bother to explain, to show like what exactly do they show. They just kind of like if they're doing and working on the same field of area, and they just repeat it again and again the same way without even showing how exactly, and we need to believe this without understanding like what is behind this data. I still don't understand how they do this panel A. So all in all, I'm very sorry, but like dear authors who will maybe listening to that, I'm convinced that there are multiple connections with multiple things. Yeah, so either you take genome, metabolism, microbiome, and there are some correlations. But I knew that already before. Like this is nothing new for me. So, and like what they show that there could be some omics data or some bacteria or some metabolites which could predict the insulin resistance. This is also not something new for me. So I don't know how they managed to publish in Nature, I should say, and I'm not very. But as I said, so I'm not very knowledgeable in that particular field. I'm just saying that as someone from outside of the field, that my personal interpretation of this paper is not very high, as also Alex mentioned. So I agree with her. So not very convincing, I should say. Yeah. But what I wanted to say about Shazia and about like Huma and also Dr. Awan and everyone in their group, thank you very much, because I think this is not an easy one, really not an easy paper to discuss, but I think you did it very well, and I liked it. I enjoyed it very much, and this was the first journal club in this format for you. So I would like to thank you. I think it is really great how you did that. Yeah. Anyone else would like to add anything here?
Yeah, I would like to say that Shazia, your TPT was very nice and very well structured, so I did also like it a lot. Thank you.
Thank you very much, Elia and Alex and your team for giving us this platform, and then confidence to all of us, especially Shazia, for taking courage in presenting this rather complex paper. Even we have learned many things, and with your discussion, there are many more questions that now we have to ponder about this, that how a study could be better designed and how the interpretation of results could be done. So indeed, it was a great learning experience for all of us, my students and my colleagues. And thank you very much for giving this platform. Your feedback, your input, your criticism was absolutely marvelous for us. Thank you very much. So, Shazia.
Thank you everyone. Any other comments?
Thank you so much.
Thank you.
Okay. Then I wish everyone to have a great day, and we will meet each other in 2 weeks, and that will be Donfa, who will present his next journal club, and we will upload the video in 1 week, and then we will be waiting for your comments. Okay.
Yes, sure. We'll see you next time.
Okay.
Thank you.
Yeah,
Thank you.
so much.
Thank you very
So
much.
much.
Bye bye.